Hi everyone, today we're going to be solving AQA GCSE Biology Foundation Tier Paper 1F. In this particular video, we are solving Specimen 2018. We're going to be solving the entire question paper in this video. Figure 1 shows the diagram of a human heart. We can see the pulmonary artery is labeled and it asks that what is section A. First, let's see what are the sections we have here. First of all, we have aorta. Now the aorta comes from the left ventricle, so this is the aorta. Then we have the upper portions, the two chambers, which are the atriums. Now, since the heart is a mirror image, so this will be the right atrium and this will be the left atrium. And the bottom portion will be the ventricle. So in this case, this will be the right ventricle and this will be the left ventricle. All right. So the question says which part of the heart is labeled A. So we already know which one is the aorta, which one is the atrium. So yes, none of them are the correct answer. Okay. And then about the valve. These are the valve. This is bicuspid valve. So we will call it atrioventricular valve. This is tricuspid valve, and on here this is the bicuspid valve, but we will in general name it atrioventricular valve. These are semilunar valve. So the main thing is they're all valves. Okay, they prevent backflow of blood. Now our answer will be ventricle. A is ventricle. Where does the pulmonary artery takes blood to? Anything related to pulmonary means lungs, and artery takes blood away from the heart. That means it will take the blood to the lungs. Circle a valve on figure 1. So we have already seen there are many valves here. This is a valve, this is also a valve, this is also a valve, and this is also a valve. So technically we can just circle any one of them. The coronary arteries supply blood to the heart. Figure 2 shows two coronary arteries. A healthy artery, where we can see the lumen is very wide in the artery wall, and artery affected by coronary heart disease with fatty deposit. You can see the blood flow is very constricted. Describe two ways the healthy artery is different from the artery affected by coronary heart disease. From the picture, we can see the healthy artery has a very large lumen, and there are no fatty deposits on that particular lumen. Since it is more wider, so more blood will be able to flow through the healthy artery. So we will answer in this way. There are no fatty deposits and healthy artery is wider, so more blood can flow through it. On the contrary, the unhealthy, the artery that is affected by coronary heart disease, or the unhealthy artery in this case, all right, we see blood flow restricted, and also we can see fatty deposits, which is the main cause of the restriction of the blood flow. What can be used to treat people with coronary heart disease? People with coronary heart disease require some form of medicines that will allow that particular blood vessel to widen up. Antibiotics are used for, it is a medicine that is used to fight bacteria. Hormones are used, a medicine, all right, where there is deficiency of hormones. Vaccination is done to give a, basically ability to produce our own immunity against a particular virus or a pathogen. So in this case, statins and stent will be the answer. Statins are a type of drug that can decrease the fatty deposits, and stent is a mesh, it's a wire mesh that can, physically widen the artery so that more blood can flow through it. Suggest two risk factors for coronary heart disease. The question asks for two risk factors. Well, for coronary heart disease, there are many things that can be done that can increase the risk factor for the chance of having more coronary heart disease. They will be smoking, having a high-fat diet, and having no exercise or very little exercise in day-to-day life, being very overweight, and having high blood pressure, and also having high cholesterol level. All of these will lead to coronary heart disease. Figure 3 shows the percentages of adults in UK who have coronary heart disease. You can see the males and females. We notice a general trend is that as the age increases, the amount of coronary heart disease is also increasing. However, we also notice another trend, which is males generally have a higher percentage of coronary heart disease than female. Calculate the difference in the percentage of male and female adults aged 65 and over who have coronary heart disease. So we can see from the bar chart that 10% of female have coronary heart disease, whereas 18% of male have coronary heart disease. We will find out the answer by subtracting 10 from 18. So 8% more males have coronary heart disease compared to female, which is the correct conclusion for the data in figure 3. Okay, children do not suffer from coronary heart disease. We notice that coronary heart disease information starts from 18 and above 54. So there are no data here present. So we can't say anything. Cannot conclude anything about it. More males suffer from coronary heart disease than females. Yes, we have seen that higher percentages of male have coronary heart disease. So this is correct. Then more younger people suffer from coronary heart disease than older people. No, higher percentages of older people have coronary heart disease. So the second option is the correct answer. Catalase is an enzyme. Catalase controls the following reaction. When hydrogen peroxide is broken down by the help of the enzyme catalase, it turns into water and oxygen. By the way, guys, you need to understand one thing. Hydrogen peroxide is often the byproduct of respiration. And if you have ever used hydrogen peroxide, you know that this hydrogen peroxide has a bleaching properties or a oxidizing properties. Okay, we can say it has a oxidizing properties. So because it has that particular property, it can be very dangerous if the concentration of hydrogen peroxide within the body increases too much. So catalase enzyme helps to fight it back. A student did an investigation on catalase activity. This is the method used. Put 1 cm cube of hydrogen peroxide solution in a test tube. Add 1 cm cube of catalase solution. Bubble of oxygen are produced. Bubbles cause foam to rise up the tube. Measure the maximum height of the foam. Figure 4 shows the experiment. We can see the experiment, foam rising up the tube. 1 cm cube of catalase solution and 1 cm cube of hydrogen peroxide is put into this experiment and the height of the foam is measured. The experiment was carried out at 20 degrees Celsius. Okay, so the height of the foam is indicating what amount of oxygen is produced in this experiment. The table 1 shows some result from the investigation. At 10 up to 60 degrees Celsius, we can see the activities. Alright, if we notice is that at higher temperature, as the temperature increases above 30 to 40 degrees Celsius, we can see maximum height of the foam has decreased consistently after that. That means the enzyme is basically, getting denatured or destroyed, which is why it's not working anymore. So it works much better at 30 degrees Celsius. Why did the student carry out the experiment 3 times at each temperature? To make the experiment more accurate. Well, it can improve the accuracy of the experiment by taking average results and, eliminating anomalous results. However, it doesn't make the experiment accurate. The experiment is okay by itself. Repeating 3 times actually tries to show that the experiment is more repeatable. To prove the experiment was correct? No. To show the experiment was more repeatable. Correct. So that the experiment is more repeatable and it can give a consistent result. Alright, we can see it can give a consistent result. So it shows that it is more repeatable. The student thought one result was anomaly. Circle the anomalous result in table 1. When trying to find out an anomalous result in a table, we have to understand that an anomalous result is the one that doesn't match the trend. So for example, at 10 degrees Celsius, if we search, they are more or closely related values. Whereas at 20 degrees Celsius in test 1, we only have 0 height of the foam. That means this must be an error. Whereas the other 2 tests has 3.3 and 3.1. So temperature of 20 degrees Celsius, test 1. What did the student do with the anomalous result? Whenever there is an anomalous result, the student will not use it for calculating average. So the student ignores it. What conclusion can be made as the temperature increases? Okay, we can see point number 1, decrease the reaction rate up to 30 degrees Celsius. No, we know that we have seen from the data that the reaction rate was increasing up to 30 degrees Celsius. Decreases the reaction rate up to 40 degrees Celsius. So this shows for wrong statement, because up to 30 degrees Celsius, the rate of reaction was increasing, and then from 40 degrees Celsius onward, it decreased. Increases the rate of reaction up to 30 degrees Celsius. So this is the correct incorrect statement. Increases the rate of reaction up to 40 degrees Celsius. This is wrong. This is wrong because we can see that at 40 degrees Celsius, the experiment produced less volume or less height of the foam in comparison with 30 degrees Celsius, which has a higher height of the foam. At which temperature was catalase denatured? So we can nicely say at 60 degrees Celsius, catalase was denatured. Denatured meaning that this particular enzyme has changed its shape permanently and it went to the point of no return, that the shape of the enzyme will ever be back. Okay, it's like having an egg white and then boiling it. If you were to boil an egg white, then it will turn into the clear liquid will turn into white, and that indicates you will never be able to go back to that white in the clear liquid back again. So the similar thing happens with the enzyme. Enzymes are protein, they get denatured, and the denaturation happened at 60 degrees Celsius because we could see that at 60 degrees Celsius there was no reaction. The student thought the optimum temperature for catalase activity was between 30 degrees Celsius and 40 degrees Celsius. How could the investigation be improved to find a more precise value for the optimum temperature? When a student finds out that the highest rate of reaction occurred at 30, but then the rate of reaction decreased at a higher temperature like 40, then the student can repeat the experiment in between the temperature of 30, then 32, 34. In that way, it can go up until 40 degrees Celsius. Repeating the 30 degrees Celsius and 40 degrees Celsius is not important because this experiment was already done. All right. So can choose the intervals and repeat it. Do the experiment at 70 and 80. So that will be wrong because, the experiment needs to be repeated between 30 and 40. Do the experiment at 30, 35 and 40 degrees Celsius. This could be a better approach compared to the top one. Use less hydrogen peroxide in solution. No, hydrogen peroxide must be kept the same volume and same concentration. Use more catalase. No, same amount should be used. So repeat the experiment at more intervals. Amylase is the enzyme that controls the breakdown of starch to glucose. Describe how the student could investigate the effect of pH on the breakdown of starch by amylase. Since in this particular experiment, we are trying to investigate the effect of pH, so we must use a range of pH in that particular experiment. Now, we must understand living things operate at a certain pH. For example, consider our human body, all right? In our human body, we have generally our mouth, which is generally close to pH 7. It can be slightly acidic depending, all right? However, in the stomach, it is very acidic. And then in the large intestine, it is alkaline, like 8, pH 8. So we can use a range of pH. For example, we can use pH of, let's say, pH 2, pH 4. We can repeat the experiment at pH 6, then pH 8 or pH 10. These are the pH values that we can repeat the experiment. Now, how would you keep the pH, values, fixed in an experiment? We would use a buffer solution, all right? After that, we will keep the other things, all right, similar, constant, all right, to have a fair experiment. For example, we'll keep the amount, which generally means the concentration of the starch and the amylase same, meaning we're going to keep the volume and the concentration of starch and amylase same in all experiments. We'll keep the temperature same for all of the experiment using a water bath, because here we are only testing the effect of pH, so we will have to keep the temperature same for every experiment. So let's say we will repeat the experiment at a human body temperature, which is 37 degrees Celsius. So all of the experiments will be done at 37 degrees Celsius. Okay, and we will maintain it using a electric hot water bath. Then we are going to use iodine to test, all right, to test for the presence of starch, as the amylase is going to work on that starch, is going to break down the starch. If we test the solution, if we take at an interval, like every minute, if we take a drop of that particular solution from the test and then put it in iodine, and we see that there is a blue-black color that will indicate the presence of starch, then what we're going to do is that we are going to observe the color changes at different temperature, all right? We will repeat the experiment at each pH, all right? Once we repeat the experiment at each pH, then we will get a, clear understanding of how how the enzyme is affected by the changes in pH. All right. So the answer can be written as follows. Figure 5 shows a human chick cell viewed under a light microscope. Label the nucleus and cell membrane on Figure 5. The nucleus will be the most darkly stained portion. So definitely this will be the nucleus. And the cell membrane will be the outer edge. Cheek cells are types of body cells. Body cells grow through cell division. What is the name of this type of cell division? Cell division that produces identical cells are called mitosis. Ribosomes and mitochondria are not shown in Figure 5. What type of microscope is needed to see ribosome and mitochondria? Because mitochondria and ribosomes, they are very, very small. So ribosome, first of all, it has the size of 25 nanometer. To see 25 nanometer, we will need an electron microscope. Okay, so for those who, you have forgotten, all right, light microscope or LM has a maximum resolution of 200 nanometer. Anything smaller than that will not be visible in light microscope. Whereas a EM has a maximum resolution of 0.5 nanometer. Some of them can even go even smaller than that. So, we can see very nicely ribosomes in electron microscope. But with light microscope, we will not be able to distinguish. What is the advantage of using the type of microscope you named in part 3.3? All right. In part 3.3, we have mentioned electron microscope. Electron microscope has very high magnification. We can magnify it to a very high level, and yet we can see a very clear picture. So, high magnification. The cheek cell in figure 6 is magnified 250 times. The width of the cell is shown by the line D to E. You can see from D to E. Okay. Calculate the width of the cheek cells in micrometers. The width of the cheek cell provided in the answer is 45 millimeter. In the exam, we will measure it using a ruler. However, in an electronic device, it's not possible to measure. So 45 millimeter. 1 millimeter is equals to 1000 micrometer. So we will use the equation to work out the real width of the cell in millimeter. The real size is equals to image size divided by magnification. So the image size is 45,000 micrometer divided by magnification, which in this case, the magnification is 250 times. So we will divide it by 250. This gives us, so they want the answer in millimeter first. So 45 divided by 250, the answer is 0.18 millimeter. And then we will convert that into micrometer. 0.18 times 1000 give us 180 micrometer. A red blood cell is 8 micrometer in diameter. A bacterial cell is 40 times small. Calculate the diameter of the bacterial cell. So, since 8 micrometer is the diameter of the red blood cell, if we divide it by 40, then we should get the size of the bacteria. So 8 divided by 40 gives us 0.2. Since the calculation started with the unit micrometer, so 0.2 micrometer will be the answer. Microorganism can cause disease. Draw one line from each disease to the correct description. So HIV, we know that HIV is caused by a virus. So let's see, HIV can spread by not washing hands thoroughly. No, we know that salmonella can spread by not washing hands thoroughly, because salmonella is a type of bacteria that is a foodborne illness, and not having a washed hand will give us salmonella poisoning, food poisoning, or salmonella. HIV, all right, can increase the chance of infection such as pneumonia. When HIV turns into AIDS, then it increases the chance of, opportunistic infections such as pneumonia. Normally a person will not get pneumonia. Generally, the immunity will protect the person. However, a person with HIV can easily contract pneumonia when the person contracts cold. Then when talking about malaria, part of the life cycle includes an insect. This is correct. Malaria includes a mosquito, a female Anopheles mosquito. So malaria will involve that. Now, why the other points are not correct? It says spread by coughs and sneezes. When talking about spreads by coughs and sneezes, then we can talk about common cold or flu that spreads by, coughs and sneezes. Treated with stem cells, these are usually genetic disorders. Treated with a fungicide. To treat something with a fungicide must be a fungal infection. Gonorrhea is a sexually transmitted disease. A bacterium causes gonorrhea. What are the symptoms of gonorrhea? All right. So it says that we have to tick 2 boxes. So whenever you're asked to tick 2 boxes, make sure that we tick 2 boxes. Otherwise, the answer will be wrong. If we tick 3 boxes and 2 of the boxes are correct, we'll still get only 1 mark. And if we tick only 1 box, then you already get it. You won't be getting second mark anyway. So, headache, pain when urinating. So, gonorrhea is a sexually transmitted disease. So, it will definitely lead to, because it is a disease of the urinary tract system, all right, and it will cause infection in the urinary tract system. So, there will be pain during urinating. However, there will be headaches, all right, directly related to gonorrhea. Then there will be rash, vomiting, and yellow discharge. Yellow discharge is the correct point for gonorrhea. Vomiting and rash are not part of gonorrhea. They are part of other diseases. For example, rash can be from measles, all right. Headache can be from fever, all right, or vomiting. It can be induced due to food poisoning. So they are not the symptoms for gonorrhea. Table 2 shows the number of people in the UK diagnosed with gonorrhea in different years. Year 2005, female 5.0, number of people diagnosed with gonorrhea in thousands. All right. So we can see from 2005 to 2013, the number of female treated increased and the number of male treated increased. However, the number of male, all right, is always higher than female. So we can definitely understand that this particular disease affects male more than female. Use the data in table to complete figure 7. The number for males have already been plotted. Only some of the numbers for females have been plotted. Okay, so describe the pattern in numbers of males and females with gonorrhea from 2005 to 2013. We can see for both males and females, up until 2005 to up until 2009, the level remains fairly constant. However, from 2009, the number of people, specifically males, contracting gonorrhea increased very high. Whereas in females, it rised, but not as much. Males rises faster than the females. So, we will write the answer in this way. The number of males and females with gonorrhea from 2005 to 2009 is fairly level. However, there is a rise after 2009. Males are always higher than females in contracting gonorrhea. From 2009, the males contracting gonorrhea rises faster than females. Gonorrhea is treated with an antibiotic. HIV is another sexually transmitted disease. Guys, HIV is caused by a virus and gonorrhea is caused by a bacteria. So that can understand why we are treating it with antibiotics. Explain why prescribing an antibiotic will not cure HIV. So we will write the answer in this way. HIV is caused by a virus and antibiotics are only effective against bacteria. Antibiotics do not kill viruses. Anaerobic respiration happens in muscle cells and yeast cells. The equation describes anaerobic respiration in muscle cells. So from glucose, it produces lactic acid. How can you tell from the equation that this process is anaerobic? When lactic acid is produced from glucose in anaerobic respiration, it does not use any oxygen. So that's how we can say that this process is anaerobic. Anaerobic means without oxygen use. So the answer will be no oxygen is used. Exercise cannot be sustained when anaerobic respiration takes place in muscle cells. Question is, explain why. If a particular exercise leads to anaerobic respiration in muscles, the muscles easily become fatigued, and as soon as the concentration of the lactic acid within the muscle increases, it will stop contracting because the muscle is not provided with enough energy to contract. Figure 8 shows an experiment to investigate anaerobic respiration in yeast cell. We can see to do the experiment, liquid paraffin is put on top of the yeast cell in sugar solution so that the yeast cell does not get any supply of oxygen. And then whatever gas is collected, it is then bubbled through a lime water so that we can, react to the carbon dioxide with the lime water and we can see that the CO2 is being produced. What gas will bubble into tube B? So definitely anaerobic respiration of yeast will produce carbon dioxide. So it will be carbon dioxide. Describe how you could use the tube B to measure the rate of the reaction in tube A. We can do it in two ways. First of all, either we can count the number of bubbles that are produced in a given amount of time, or we can measure the volume of gas that is produced in a given amount of time. Anaerobic respiration in yeast is also called fermentation. Fermentation produces ethanol. Give one use of fermentation in the food industry. So we use fermentation for making bread, or we do it for brewing, wine and liquors. So we will write bread brewing, bread making, and brewing. Plants have adaptations to help defend themselves and to help them survive. Figure 9 shows a nettle plant. Alright, explain how the nettle is adapted for defense and protection. From the diagram, we can see that the nettle plant contains some spiky hairs, alright, which can sting. So that when herbivores animals tries to eat them, alright, they will get sting in their mouth, and as a result, less amount of the plant is eaten by the herbivores. So the answer can be written like this. Nettle plant contains stinging hairs, and these hairs harms the herbivores when the herbivorous animals wants to eat them. So less amount of the plant is damaged by herbivorous animal, as the animal soon gets sting from the nettle plant and stops eating. Witch hazel is another plant adapted for defense. Witch hazel produces oil with antiseptic properties. The oil prevents bacteria from attacking the plant. Our students investigated how effective 3 different plant oils were at preventing the growth of bacteria. You can see in an agar plate, we have paper disc that is soaked with clove oil. Then we have paper disc that is soaked with lemon oil, and paper disc that is soaked with hazel oil. We can see that the paper disc soaked with clove oil has the highest clear region, meaning that clove oil is the best at killing bacteria. However, paper disc soaked with hazel oil has a second highest clear region, meaning it is the second best. So it will be at the second position. This will be at the first position. And lemon oil is not effective in killing bacteria. Which oil plant is the most effective at preventing the growth of bacteria? Give your reason. So the oil in this case would be clove oil, and the reason will be because clove oil has the largest area with no bacteria growing in it. The student tested tea tree oil using the same method. The results showed tea tree oil was the most effective at preventing bacterial growth. The student concluded that tea tree oil could be used to treat bacterial infection instead of antibiotics. Give one reason why this is not a valid conclusion. For this experiment, if we look back into this particular agar plate, in the agar plate, we see no antibiotics being tested. So there are no antibiotics tested with the paper disc that are soaked in different tree oils. So in that case, all right, the antibiotics were not tested. That's why this particular conclusion is not valid. After a meal rich in carbohydrate, the concentration of glucose in the small intestine changes. Here we can see a table where it shows the concentration of glucose in mole per dm cubed at different distances in the small intestine. We can see 100 centimeter to 700 centimeter distance, and we can see the concentration initially increases, then decreases to 0. At what distance the small intestine is the glucose concentration highest? So we can see at a distance of 300 centimeter, it has 500 mole per dm cube. So definitely 300 centimeter will be the correct answer. Use the data in table 3 to plot a bar chart on figure 11. We have to label the y-axis and choose a suitable scale. So the x-axis is done for us, the distance along intestine in centimeter. Now, we need to look back into this particular table. In the table, the concentrations ranges from 0 and maximum is 500. Then we will go back into the graph and we will see that, let's say if 2 boxes is equals to 50, then we have 100, 150, 200, 250. That means we cannot reach 500 here. So we cannot use this particular scale. If we use 2 boxes is equals to 100, then we have 200, we have 300, then we have 400, 500 and 600. All right, now this concentration in this case is given in glucose in mole per dm cube. So on the y-axis, we will say concentration of glucose in mole per dm cube. Describe how the concentration of glucose changes as the distance increases along the small intestine. If we look into the particular bar chart, we see as the distance increases from 0 to 300, the concentration of glucose goes from 0, let's say, to 500 mole per dm³. So we can conclude that in the description. Also, we can say that as the distance goes from 300 to 700, the glucose concentration decreases from 500 mole per dm³ to 0. So the answer will be from 0 to 300 centimeter, the glucose concentration increases from 50 to 500 mole per dm³. And then from 300 to 700 centimeter, the concentration of glucose decreases from 500 to 0 mole per dm³. Explain why the concentration of glucose in the small intestine changes between 100 centimeter and 300 centimeter. So basically, in the first portion of the small intestine, the carbohydrates are still getting broken down, and they are getting broken down into sugars, simple sugars, which are glucose. And the broken down, of the breakdown of carbohydrates is actually occurring by an enzyme. The enzyme is amylase in this case. Explain why the concentration of glucose in the small intestine changes between 300 centimeter and 700 centimeter. So from 300 centimeter to 700 centimeter, the absorption of glucose is occurring into the blood, and the absorption is mainly taking place by active transport against the concentration gradient, which is why the concentration of glucose goes from 500 mole per dm³ to 0 mole per dm³ within that distance. To be healthy, plants need the right amount of mineral ions from the soil. In figure 12, we can see 4 plants. The plants are grown in 4 different growing conditions. We have sunny area with nitrate and magnesium added to the soil. Then in our plant B, we have sunny area with magnesium but no nitrate added to the soil. So guys, when nitrate and magnesium is present, plants are able to make chlorophyll. Chlorophyll is a protein but requires magnesium. When magnesium is present but no nitrate is present, the plants are unable to make protein. So we need to keep that in mind. In sunny area with nitrate but no magnesium, again guys, this plant is able to make protein, but because chlorophyll requires magnesium, so less chlorophyll. The reason why we will have less chlorophyll, because the soil by itself will have some magnesium in it, but added magnesium gives the plant a boost to grow. Plant growing in dark area will not be able to do photosynthesis, thereby it will lack the energy to grow. Even though nitrate and magnesium is available, and they will not be able to use it that much. Okay, so in plant D, we can already see there are yellow patches, meaning there is a lack of chlorophyll. Which plant was grown with no nitrate? Guys, in the diagram, we can see plant A is a little bit smaller than all other plants. We can see plant B, it's quite big with broad leaves. All right, plant C is quite tall, okay, and plant D has yellow patches. So from this, we can conclude that plant C has grown in dark. Because the plant is growing in dark, so it is trying to grow as tall as possible to reach the sunlight. Whereas the plant with yellow patches, it could be the plant that has no nitrate. All right, so no nitrate will be plant A, because we can see that the plant A is smaller. Lack of nitrate meaning the plant will not be able to make protein. Okay, so the plant will have a stunted growth. And the plant with yellow patches will be definitely with the one with no magnesium, because as I told you, magnesium is needed for chlorophyll. And plant B is the one that is going to be with everything in it. So plant B will be sunny area with a nitrate present and magnesium added to the soil. The question says which plant was grown with no nitrate. So definitely a stunted growth will be an indicator of no nitrate. Which plant was grown with no magnesium? Since there is less chlorophyll in plant D and it is becoming yellow, that means plant E has no magnesium in it. Give one variable that was kept constant in this experiment. So in this experiment, they used the same type of plant for all of the experiment. Another thing that must be kept same in all of this experiment is that all of the plants should be given equal amount of water to grow. Plants need other minerals for healthy growth such as potassium ions and phosphate ions. A farmer wanted to compare the percentage of mineral in two types of manure. Cow manure from her own farm, chicken manure pellets she could buy. Table 4 shows the data for each type of manure. So when it comes with cow manure, the phosphate ions is 0.4 and potassium ions is 0.5. But with chicken manure pellets, the phosphate ions is way more higher. We can say almost 5 to 6 times higher, and the potassium ions is almost 5 times higher. Suggest one advantage and one disadvantage of using the chicken manure pellets compared to the cow manure. Guys, the first idea in terms of advantage that comes with chicken manure pellets is that it gives more mineral ions to the plant. However, the disadvantage is that the farmer needs to buy it, whereas the cow manure is free. Plants transport water and mineral ions from the root to the leaves. Plant move mineral ions from a low concentration in the soil to a high concentration in the root cells. What process do plants use to move these mineral ions into root cells? Because the plant is moving from the low concentration to a place of high concentration, that means an active transport is needed. So guys, what is active transport? Active transport is the movement of ions against the concentration gradient using energy from respiration. So farmer will use active transport in this case. So the root cell will use active transport in this case. Describe how water moves from roots to the leaves. During the day, when the tree are exposed to sunlight, the stomatas are open. So what happens is that water vapor from the leaf evaporates and it goes into the atmosphere. This causes the cells within the leaves to have a high concentration gradient, which means that the leaves have low water potential. The low water potential or a high concentration in the leaf cells, it acts like a suction pool on the xylem, and that drives all the water that is taken up by the root hair cells into the xylem and into the leaf cells. So the answer can be written as the water moves from the roots to the leaves by transpiration pool in xylem. Plants lose water through the stomata in the leaves. The epidermis can be peeled from a leaf. The stomata can be seen using a light microscope. Table 5 shows data a student collected from 5 areas on a leaf. Number of stomata, we can see the upper surface, the lower surface. Alright, so in the upper surface, we can see there is very few or even no stomata. Average we can see 2. But the lower surface is the highest number of stomata, which is consistent near to 40. Describe how the student might have collected the data in table 5. Guys, this is a microscopy related question. So you can see the epidermis can be peeled from a leaf. We know that a general plant structure consists of a epidermal layer, and then underneath that we have palisade mesophyll, and underneath that we usually have a vascular bundle which consists of xylem and phloem. The phloems in the diagram are mentioned in red, and in between them usually we find a particular layer called cambium. Okay, so the xylem are at the top. Now underneath this we usually find a spongy mesophyll cell layer, and underneath that we can find out the lower epidermis with guard cells. Okay guys, now the question says that the student peels the leaf. Okay, the epidermis can be peeled from a leaf. The stomata then can be seen using a light microscope. So we can peel this epidermis layer, all right, and then we can see the stomata. We can also do the same for the top part. So there must be stomata present at the top part as well, because you can see the top part also has stomata. The upper surface has stomata, but less in number. So how will the student collect the data? First of all, the student will mount the epidermis on a slide. Then the student will count the number of stomata within the field of view and repeat the same process for 4 more areas within the same sample, and then repeat the method on other surface of leaves, like the opposite surface, which is like, for example, either the lower epidermis or the upper epidermis, and then for each surface the student will calculate an average. What is the median number of stomata on the upper surface of the leaf? If we arrange the data from the upper surface of the leaf, we get 0, 1, 1, 2, 3, and we get 0, 1, 1, 3, and 5. So the median comes to be 1. Median is the middle of all of the numbers, so it's 1. Calculate the value of x in table 5. Give your answer to two significant figures. To calculate the value of x in table 5, we may need to reject any anomalous result. However, in this data, we do not see any anomalous result. So we will do 44 plus 41 plus 40 plus 42 plus 39, and then we will divide by all of the samples. The answer is 41.2. However, we have to give the answer in two significant figures, so the answer will be 41. The plant used in this investigation has very few stomata on the upper surface of the leaf. Explain why this is an advantage to the plant. Having less stomata in the upper surface of the leaf can be an advantageous part of the plant because less amount of water vapor will be lost through the upper surface of the leaf. This means that the plant will not wilt even if it absorbs less amount of water from the soil. So the answer can be written like this: less amount of water is lost and the plant does not wilt. Tobacco mosaic virus or TMV is a disease affecting plants. Figure 13 shows a leaf infected with TMV. We can see yellow patches where TMV has destroyed the chloroplast and they grew in number. All tools should be washed in disinfectant after using them on plants infected with TMV. Suggest why? Because TMV is a virus, so that we can kill all the virus and it does not spread the virus to other plants. Scientists produced a single plant that contained a TMV-resistant gene. Suggest how scientists can use the plant to produce many plants with the TMV-resistant gene. Scientists can take stem cells from the meristem of this particular TMV-resistant plant, and then the scientist can perform tissue culture on that particular meristem, on the stem cells of the meristem, and produce many genetically identical plants, those that are resistant to the TMV virus. Some plants produce fruits which contain glucose. Describe how you would test for the presence of glucose in fruit. Glucose can be tested using Benedict solution because glucose is a reducing sugar. So if we use a Benedict solution and we heat the Benedict solution along with the fruit juice, then we will see that the glucose solution will turn blue to orange. The Benedict solution has a blue color, which will then turn to orange, indicating the presence of glucose. TMV can cause plants to produce less chlorophyll. This causes leaf discoloration. Explain why plants with TMV have stunted growth. Because the plant with TMV produces less amount of chlorophyll, the plant can do less photosynthesis. And as a result of that, the plant produces less amount of glucose, which is needed mainly for energy release for growth. And glucose is also needed for, respiration. Glucose is also needed in plants as a building block, for making cellulose. And plant converts glucose into amino acids, proteins. So if there is less glucose, then less amino acid is produced and less proteins are available for growth. And because glucose is also needed for TMV, can cause plants to produce less chlorophyll. This causes leaf discoloration. Explain why plants with TMV have stunted growth. So TMV virus, since it doesn't allow the plant to produce chlorophyll, because there is less chlorophyll, there will be lack of glucose. And a lack of glucose will lead to less amino acid and cellulose. And lack of amino acid will lead to less protein for growth. So by keeping this into understanding, also we need to understand that glucose is needed for energy. So how we are going to write down the answer? The answer is going to be written like this: less photosynthesis because there is less chlorophyll, and therefore the plant is able to make less glucose, so less amount of energy can be released from that particular glucose for growth. And lack of glucose also leads to less amino acid, less cellulose, and less protein to be made within the plant, which are very much needed for the plant's growth. Microorganisms cause infections. The human body has many ways of defending itself against microorganisms. Describe 2 ways the body prevents the entry of microorganisms. First of all, we have to understand what are the barriers for microorganism within our body. First of all, if we look into the first biggest barrier is the skin. And what does the skin do? The skin produces antimicrobial secretion in the sweat, which kills off the bacteria, specifically the bad bacteria. Then we have hairs in the nose that traps any particles that contains pathogen. Also, the trachea has mucus which can trap pathogens within that mucus, and the mucus will then kill that particular pathogen. Then, if any bacteria or any microorganisms are swallowed, the stomach contains acid which kills the microorganism within the food. We will have to only give 2 points. However, I will mention you the 4 points in the answer. In 2014, the Ebola virus killed almost 8,000 people in Africa. Drug companies have developed a new drug to treat Ebola. Explain what testing must be done before the new drug can be used to treat people. To use a new drug for treating people, preclinical trials of the new drug must be done. So the preclinical trials are done on live animals. It is done to test the toxicity and the dosage requirement and the efficacy of the particular drug. Once it is proven that it is good enough, then the clinical trials are done on healthy volunteers and Ebola patients at a very low dosage to find out that what are the side effects of that particular drugs. And then only the trials for optimum dosage are done for the best efficacy. After that, a double-blind trial is done in which a particular group is given placebo and a particular group of Ebola patients are given the drug. No one knows who is given the placebo and who was given the drug. So, to find out that the company has made any false claims or not, then a peer review of that data is done on a different location. Once all of this process is completed, then the new drug can be released into the market. Guys, so that's all for this particular question paper. Thank you for watching the video, guys, and subscribe to the channel, guys. Question paper like this will be uploaded. We're gonna continuously upload question paper like this. And guys, if you have any suggestion, let us know in the comments, and we'll try to improve on your suggestions. And we'll make sure to put all of the videos related to this particular paper in a playlist so that you can watch them all together. Thank you, guys, for joining the video. See you in the next video, guys. Bye bye.